The cell cycle has three main phases and is constantly regulated by the three checkpoints found within. The three phases include interphase, mitosis, and cytokinesis. Interphase takes up 90% of the time and can be split up into three subsections: G1, synthesis, and G2. During interphase, the cell begins preparing to divide by growing to the appropriate size and synthesizing proteins for the synthesis phase. It then begins replicating its centromeres and DNA found within the nucleus in the synthesis phase to produce two exact chromosome copies. The G2 phase, while not necessary, is highly helpful in the division of cells. The G2 phase continues the cell's growth in preparation for mitosis, which can also be split up into four subsections. Prophase condenses all of the DNA and proteins known as chromatin into chromosomes through chromatin condensation. The sister chromatids then attach to the centromeres copied during interphase. It also dissolves the nuclear envelope surrounding the genetic material. Metaphase then aligns all of the chromosomes at the equatorial plate of the cell. During anaphase, each pair of chromosomes are separated into an identical independent chromosome. They are then attached to the centromeres by the mitotic spindles, and the sister chromatids are pulled apart to opposite ends of the cell. Telophase causes two daughter nuclei to form in each daughter cell, and then causes the nuclear envelope to reform around the genetic material. Once telophase is complete, cytokinesis divides the cell into two daughter cells by splitting the cytoplasm. A contractile ring forms around the center of the cell and begins shrinking until the two cells are pinched off of each other, forming a cleavage furrow. Each daughter cell is identical with the same genetic material as the parental cell. The G0 phase occurs due to DNA damage or degradation. This causes the cell to enter cell cycle arrest, which means until the proper conditions have been reached, the cell will not continue the cycle. The cell is regulated by three main cell checkpoints. The first checkpoint occurs within G1, which checks to make sure that the cell is large enough and that it has made the proper proteins such as p53 for the synthesis phase. The second checkpoint occurs within the G2 phase and ensures that the cell has a chance to repair any DNA that may have been damaged during replication. The M checkpoint simply checks to make sure that mitosis is complete and that the cell is ready to divide by ensuring that each chromosome is attached to the mitotic spindle. Ultimately, it is the internal and external signals that allow the cell to continue the cycle. The platelet-derived growth factor is also responsible for regulating the cell cycle by binding to the cell receptors in order to stimulate cell division in connective tissues. The maturation promoting factor, also known as MPF, is responsible for allowing the cell into the mitosis phase by removing the phosphate group added earlier. It is able to do this with the use of CDKs and cyclins. CDKs, cyclin-dependent kinases, and cyclins are other factors that contribute to the regulation of the cell cycle. CDKs promote the shift from phase to phase when bonded with a cyclin, which acts as an activator. When the cyclin binds with the CDKs, it becomes active, but once the cell has shifted phases, the cyclins degrade and deactivate the CDKs. If the cyclins fail to degrade, then the cell may enter the G0 phase and temporarily stop. If the cell cycle still cannot regulate the cell division, then cancer cells may form from uncontrolled growth and from damaged DNA. Cancer cells are cells that have mutations and no longer respond to the external factors. This leads to mutations and can eventually progress into cancer cells. The only way to stop the cancer cells from progressing any further is by using nocodazole. Last but not least, I would like to thank Mr. Anderson. Please go check out all of his videos at bozemanscience.com. Thank you.